So, hi everyone. The topic for revision for this video is antibiotics and other chemotherapeutic agents. So, first of all, talking about chemotherapy, what exactly is chemotherapy? It's the use of a chemical substance to treat a specific disease that's called chemotherapy, and the chemical substance that is used to treat the disease is known as the chemotherapeutic agent. And mostly the antibiotics that are produced commercially, they are produced by microbial biosynthesis. Antitoxins, etc., are non-chemotherapeutic in nature. The compounds that are used to kill or inhibit any kind of microbial growth in vitro, that is in laboratory conditions, they are not classified as chemotherapeutic agents. They are classified as disinfectants, antiseptics, or germicides. So these chemotherapeutic agents have selective toxicity for a parasite. And how is it done is, how is it made sure that it is selectively toxic? It has to be useful as a chemotherapeutic agent, and it has to have low toxicity for the host cell that is using the chemotherapeutic agent, while having high toxicity for the parasite against which it is being used. Germicides are non-selective in nature in terms of their action on the particular cell, and these germicides, they interfere with the natural defense mechanisms of the cell that they're acting upon. For example, phagocytosis. The germicides will be interfering with phagocytosis of the, which is the natural defense mechanism of a particular cell on which we are using this particular germicide. So the conditions that are important for an agent to be classified under a chemotherapeutic agent, the satisfying conditions that are required is, number one, that it should have selective toxicity for a particular parasite or a microbe that we or a cell that we are trying to destroy. And selective toxicity may include the destroying ability or preventing the activity of that particular parasite against which that agent is being used. Second requirement is that it is able to come in the contact of a parasite through penetration of our cells and tissues in effective concentration. The third one is that it will not affect the host's defense mechanism in any way. The natural defense mechanism of hosts are not altered through the use of that particular agent. When all these three conditions are satisfied, that's when a chemotherapeutic agent is considered a satisfactory chemotherapeutic agent. Talking about a little history of chemotherapy, where it began, how we got to know what was the first agent that was used in chemotherapy. So, Europeans in 1630s, they used to use natural quinine, from which was extracted from the bark of cinchona tree, and this natural quinine was used by Europeans in 1630s to treat the disease malaria. Syphilis is the first known disease against which a chemotherapeutic agent was used. And what was used to treat syphilis? It was mercury. Earlier, the first thing that was used to treat syphilis was mercury in 1495. After that, in the year 1910, arsenal compound, which is known as salvarsan, it was used to treat syphilis. And this salvarsan compound, which is arsenal compound, it was synthesized by Paul Ehrlich, and it was a specific drug that was used to treat the disease syphilis. Paul Ehrlich and Elik Mechnikov, they got a Nobel Prize in Physiology and Medicine in the year 1908. And why did they get the Nobel Prize? It was for the synthesis of, for the systematic and deliberate search for a compound having potential microbicidal properties, which had low toxicity for humans and animals, and it had a good chemical stability. So for this, they got a Nobel Prize in 1908. And this compound was known as Ehrlich's compound, which was basically salvarsan that was initially used for treatment of syphilis disease. Then arsenic. arsphenmine was specifically used for its action against the worm Treponema. Sulfonamides were the next compounds that were used in 1935. Domac showed their therapeutic value again, and it had non-specific uh, action, and it was effective against a broad category of pathogenic organisms. The example is Prontosil. So the sulfonamide named Prontosil, it was non-specific and it was effective against a large variety of pathogenic organisms. Followed by Gelmo in 1908, he synthesized sulfanilamide, which was the first compound in this particular group. In 1930, Eisenberg, it showed the bactericidal properties of azodies with the sulfonamide group. Then these two sulfonamides, which are basically sulfadiazine and sulfamirazine, these are the two sulfonamides that are very extensively used for their antimicrobial or antibacterial effectiveness against bacterial infection. And they have very less toxic reaction in patients. So they are hypoallergenics. So sulfonamides are used for treatment of many things. Among them are infections caused by meningococci and Shigella, the respiratory infections caused by streptococci and staphylococci, urinary tract infections caused by the gram-negative organisms. They are used for prevention of rheumatic fever. Sulfonamides are also used for bacterial endocarditis and wound infections. They are used for prevention of UTIs, post-surgery, and catheterization. So, coming on to the definition of antibiotics, what basically are antibiotics? Antibiotics are particularly those chemotherapeutic agents which we obtain from living organisms. We obtain antibiotics from living organisms, and these antibiotics are nothing but some metabolic products of some microorganisms that are released in small amounts, which upon action are detrimental or are inhibitory to other kinds of microorganisms. Antibiosis, the term was defined by Buliman in 1889. And the condition, what is antibiosis? It is basically the condition where one creature destroys the life of another creature to sustain its own life. So basically, the first creature is entirely active and the second creature is entirely passive. And there's this restriction of the life that first creature exerts upon the second creature. Waxman in 1945, he gave the present day definition of antibiotic. And what is the present day definition of antibiotic? It is so antibiotics are basically defined as those chemical substances of microbial origin which in small amounts exert antimicrobial activity. Chinese people, they used soybean curd for the treatment of boils. So, and they used to control foot infections by wearing sandals that were fury with mold. So these molds, they helped in curing the or preventing the foot infections, the bacterial foot infections. Coming on to the next thing, which is about Pasteur and Joubert. They observed that in vine cultures, the anthrax bacilli, it disappeared when there were other organisms present. In 1901, when Pseudomonas aeruginosa was injected into rabbits, they caused the protection against anthrax infection in rabbits. So, Pseudomonas aeruginosa is the present was called Bacillus pyocyanidis. Talking about bacterial antagonism. Bacterial antagonism is basically the use of lactobacilli to treat dysentery. It was defined by Mechnikov in 1899. So it is basically a type of replacement therapy where the harmless microorganism is eliminated and replaced by the one that causes disease. In 1924, Gracia and Dat, they discovered actinomycetin from actinomycetes, and this particular actinomycetin was used to lyse the bacterial cultures and for the production of vaccines. So by lysing the bacterial culture for the production of vaccines, actinomycetin was used. In 1929, Alexander Fleming was the one who contaminated Staphylococcus aureus with mold, and mold accidentally he did that. Not accidentally. So he contaminated Staphylococcus aureus with the mold and mold colony. So when the mold was, he observed a clear zone around the colony. So what was the result? He noted that it was a penicillium species named Penicillium notatum, and this species of mold, it released inhibitory substances which he named as penicillin, which was called as the miracle drug, which was the antibiotic that he discovered, Alexander Fleming. In 1939, Rene Dubos, they isolated Bacillus brevis, and they noticed that it produced a substance that killed gram-positive bacteria. So what was the substance? It was a cell-free extract that contained two active principles, the gramicillin and the tyrosine. Streptomycin was discovered by Selman Waxman and his associates. What are the major characteristics of antibiotics that are to be used as chemotherapeutic agents? These are the main characteristics of antibiotics that can be used as chemotherapeutic agents. First one is the ability to destroy or inhibit a broad spectrum of pathogenic microorganisms. Second one is to prevent the development of resistance form of the microorganisms or those parasites, basically to prevent any development of antibiotic resistance. Third one is that they should not produce any undesirable infection in the host while inhibiting the parasite. And the fourth one is that they should not eliminate the normal microflora of the human host, that it should not affect the human host. The broad spectrum antibiotics, they may eliminate normal microflora, and they do not eliminate Monnelia, which might establish from intestinal tract, it might establish an infection, and this Monnelia infection cannot be controlled by antibiotic therapy. So this is it for today. In the next video, we'll be revising mode of action of antibiotics. Hope it helps. Thank you so much.